Alright children, so now let us talk about the enzymes which help in the process of transcription. So once we talk about this enzyme, we can see how exactly the process of transcription takes place. So children, RNA polymerase is the enzyme which catalyzes the polymerization process. Now DNA polymerase plays a role in case of DNA replication. In a very similar way, RNA polymerase play a similar type of role in the process of transcription. Because here also actually a new strand is being synthesized based on the existing base sequences on the template strand. So the same thing happens here. So here RNA polymerase binds at the promoter site. So let us see that this is the DNA, this blue color is the DNA, and on this one template strand, and the starting portion is the promoter region. So somewhat here is the promoter region, right? So RNA polymerase is the enzyme here, and it is denoted as RNA polymerase at the promoter site. So RNA polymerase read the template strand actually and add complementary nucleotide continuously. Now as the RNA polymerase move along it read the strand and it continuously keep on creating the complementary strand. That is, the complementary strand is being created at the same time. For example, if on the template strand the sequence is somewhat like this, right? The sequence is like A, G, T, C, A, G, T, C, right? So as the RNA polymerase passes along it read it and also create the corresponding bases on the other side. So this is how it will keep on creating the corresponding RNA segment. Now children, what happened? The RNA is synthesized in 5' direction. Why is it so? Because if you look at the template strand, so the template strand run from 3' to 5', right? And when this synthesis happen in the anti-parallel direction, that is the 5' to 3'. So RNA is synthesized always from 5' to 3', right? Now children, we will introducing exons and introns. Why do we want to introduce it at this point? Because RNA which gets synthesized by the end of this process of transcription, we find different regions on that RNA. Later we will see. Okay, and why we are creating RNA? Because this RNA in turn can code for protein, or they can synthesize protein. Now children, exon and intron are the part of gene, right? So they are part of RNA which is being created. So talking about the exon की अगर हम बात करें तो nucleotide sequences within the gene that code for protein, right? Now it has been observed that the sequences, even if you talk about the DNA from which the RNA has been created, that DNA also, there are certain regions which can code for protein, and there are certain regions which cannot code for protein. So DNA anyways cannot code for protein, but when this DNA forms RNA, some portion of RNA can code for proteins and some portion of RNA does not code for any protein, and those portions are termed as exon and introns. exons are those portion which code for protein actually. So they actually help in the process of protein synthesis. So it is present in the mature RNA, that is RNA which is being used for protein synthesis, right? So here you see that RNA is being formed and it consists of exon and intron both. So exon are the code sequences which code for protein, and intron does not code for any kind of protein, right? So from this DNA, mRNA is produced as a result of transcription. So what I am trying to say, that as the result of transcription, initial mRNA has been produced. Now children, this initial mRNA which is being produced, this also have exon and introns, right? So exon, they code for protein, and intron, they does not code for protein, right? Finally, what happens? The RNA which we need for protein synthesis, it should code for protein because we do not want introns. So introns become undesirable in that case. So what happens, children? All the introns are now removed by the process of RNA splicing, right? So as the result of this process of RNA splicing, all the introns are now removed and the exons alone form the mature mRNA, right? And this mature mRNA is being further used for the process of protein synthesis. So, and what is that process where mRNA is used for protein synthesis? That process is known as translation, which we later discuss. That means at the end of transcription, initial mRNA is produced which has both, that is the initial mRNA which has both exon and introns. The introns are removed and finally the mature mRNA is being formed, right? So this is about the exon and the introns. Now children, let us talk about the types of RNA. So now you might be interested to understand the detailed process of transcription. But before that, it is very important to understand the different types of RNA. Because you just saw that I am using the term mRNA and you are not sure what it is. Before we get into the detail of process of transcription step by step, it is very important for us to talk about the various type of RNA and their functions with their roles. So basically there are three important categories of RNA, that is mRNA, tRNA and rRNA, right? mRNA stands for messenger RNA, T transfer RNA, R ribosomal RNA, and they are all abbreviated as TMR, right? So these are three types of RNA. Now let us look at the role of each of them. So if you talk about the mRNA, so children, it provides the template for the protein synthesis. ये जो mRNA ये क्या है? mRNA protein synthesis के लिए original strand यानी कि template strand provide करता है. So the RNA which get created as the part of transcription, that RNA is what? It is the mRNA. यहाँ पे M का मतलब होता है messenger, something which carries the information from one part to another. So this messenger RNA act as the link between DNA and protein. So it carries the message, it carries the information, that is why it is termed as mRNA. So it will actually carry the messages and carries these messages to the protein, and that synthesized protein, so that is why they are called as messenger RNA. Now the next category which we will discuss is tRNA, that is transfer RNA. So transfer means to transfer something from one place to another. So these RNA, they carries amino acids to be joined together to form the polypeptide chain, right? Now, when we say protein synthesis, how the proteins being synthesized? कैसे synthesize होते हैं protein? What are the proteins made up of? So children, they are made up of by amino acids. We have studied in class 11 also. So if I say that RNA helps in synthesizing protein, that means RNA actually bring a lot of amino acids and they join all the amino acids with the peptide bond, and that is how the proteins are formed. Okay, polypeptides are nothing but they give rise to proteins. They are proteins actually. So tRNA will carry the amino acids which need to join together to form the proteins. So that is its job. So mRNA will carry the messages from DNA. So it will decide what would be the sequence of amino acid. But tRNA, it will bring the amino acid. It will carry the amino acid so that they join together to form the polypeptide. Talking about the rRNA, that is ribosomal RNA, it is very important during translation. That means during the process of protein synthesis, so both of them will just bring in as the raw material, right? So tRNA and mRNA act as the raw material. But where exactly the process of translation takes place? That is where exactly the protein synthesis takes place. So there the role is confirmed by ribosomal RNA. So because protein synthesis takes place in ribosomes, so these RNA are present on the ribosomes. When I talk of ribosomes, ribosomes have two major subunits which is shown in your figure, which is shown in your diagram on your screen. So it consists of the larger subunit, the upper larger, and the smaller subunit, lower smaller subunit. So from ribosomes we have rRNA, right? So basically here is tRNA that carry amino acid, mRNA bring information, right? And so larger subunit of the ribosome, it acts as the enzyme, so it is known as riboenzyme, right, which catalyze the bond formation between amino acids. So you see, tRNA is bringing the amino acid here to larger subunit of ribosome. So from this larger subunit, each tRNA bring amino acid and bond formation takes place between all the amino acids. And this is how protein is being synthesized, right? So in this bond formation takes place in the larger subunit of ribosome. And enzyme riboenzyme to catalyze the reaction of peptide bond formation. So children, there are three categories we have discussed in brief. So this is mRNA, tRNA, rRNA, and it becomes easier to understand the process of transcription, right? Because we have studied about the RNA, so the process of transcription becomes easier. Okay, so now let us discuss about the process of transcription. So the process of transcription, RNA plays a leading role, we have discussed, because this helps to create the copy of RNA. Looking at the sequence, reading the sequence on template strand of DNA. So RNA polymerase is going to play the main role in the process of transcription. So the entire process of transcription involves three major stages. What are those three stages? These are initiation, elongation, and termination. So these are the three important steps by which the process of transcription takes place. Let us understand the process step by step. Now what actually happens in the initiation? RNA polymerase combine with the sigma factor and form a hollow enzyme, right? Now what is sigma factor? Sigma factor is a protein which is needed for initiation of RNA synthesis. Now when I say RNA polymerase bind to the promoter, right? So we know that promoter is nothing but the region of DNA. In that region of DNA there is the protein called sigma factor. So polymerase is actually combined with sigma factor and form the hollow enzyme. Now children, my question is, what is this hollow enzyme? we have studied in class 11 also, holoenzyme is nothing but any enzyme which is protein plus coenzyme. So these two things, enzyme plus coenzyme, they together form holoenzyme, right? So what is this coenzyme? It is the non-protein part, non-protein component which is necessary for functioning of enzyme, right? So basically here RNA polymerase or sigma factor will join together and they will form the holoenzyme. So once holoenzyme is formed, a contribution with the sigma factor, it is necessary for the function of RNA polymerase. So in that condition, the process of initiation starts. And in case of eukaryotes, some separate transcription factors are present which mediated the binding process. That means they will inform RNA polymerase about the portion is the promoter and they will bind to the promoter. So they actually act as the mediator between RNA polymerase and the promoter region. Only after certain transcription factors are attached to the promoter, RNA polymerase will be able to bind into it. So in case of eukaryotes, we have a couple of transcription factors which act as mediator for the binding of RNA polymerase to the promoter region. But otherwise, in the process of initiation, basically what happens is RNA polymerase bind to the promoter region of DNA. But how it will bind to the promoter region? There are some factors known as sigma factors which are basically very essential for the functioning of RNA polymerase, right? So somehow at the promoter side, the sigma factor with the RNA polymerase, they join and start producing the RNA. Now in the next step is elongation. What happens after when RNA polymerase has bind to the promoter region? RNA polymerase will facilitate the opening of helix. So here children, right now DNA is like tightly bounded double helical structure and we need only one strand of DNA to act as the template strand for the synthesizing RNA, right? So for that purpose we need to unwind the two strands of DNA, right? So how it will happen? The double strand of DNA becomes unwind as the RNA polymerase move further. It keep on opening two strand of DNA, right? And once the RNA polymerase reach to the terminator end, what will happen? Again, the two strands will separated by this enzyme. Again start binding each other, right? So RNA polymerase travel the template strand. So RNA moves in the direction, it travels 3' to 5' direction. क्योंकि टेम्पलेट स्ट्रैंड क्या होता है? टेम्पलेट स्ट्रैंड 3' से 5' डायरेक्शन में होता है. तो दैट इज व्हाई आरएनए पॉलीमरेज मूव्स from 3' to 5'. So the copy of RNA is created using the complementary base sequences, right? तभी तो this mRNA which is created is exactly similar to the coding strand, है ना, क्योंकि complementary base sequences होती हैं. So elongation also involve proofreading to replace the incorrect match bases, right? So sometime it might happen that the RNA polymerase might encounter an error, right? So it might commit a mistake, means incorrect pairing has been done. So there is the provision to proofread and correct the mistake. But DNA proofreading is more efficient than RNA proofreading because of the double-stranded structure of DNA. तो ये process जो होती है ये elongation कहलाती है, right? Now the next step of this process is termination. So what will happen here? RNA polymerase travels and finally it encounter with the rho factor, right? तो finally जो है ये rho factor के साथ जुड़ जाता है. Rho factor terminator factor कहलाता है. Now children, as soon as RNA polymerase come in the contact of rho factor, it change the functionality of RNA polymerase. So RNA polymerase get information that it is okay, it's time to stop the process of transcription, right? So this rho factor disestablish the interaction between template strand and newly formed RNA, जैसे ही RNA पॉलीमरेज टर्मिनेटर तक पहुंचता है, इट गेट द इंफॉर्मेशन टू स्टॉप द ट्रांसक्रिप्शन. उसके साथ ही कनेक्शन बिटवीन टेम्पलेट और न्यूली स्ट्रैंड RNA, जो कनेक्शन होता है बिटवीन द टू स्ट्रैंड्स इज लॉस्ट. As a result, RNA come out of this portion. So newly formed RNA falls off. Where will it go now? Just for your information, where this entire process takes place. This entire process takes place in DNA. And DNA is where? Inside the nucleus. So inside the nucleus, replication takes place. Transcription also takes place at the nucleus. But this RNA which is being produced, it come out from elongation complex but still remain in the nucleus. After certain processes, the mature mRNA finally moves out from the nucleus into the cytoplasm, right? So that is how the entire process happens. So this is the process of transcription. So simply it happens in the prokaryotes and it also happens in the eukaryotes, right? Now about the transcription process. Now whatever we have discussed is in general for any transcription process, but children, it has been observed that in case of eukaryotes there are certain additional complexities which are experienced, right, which are not present in the prokaryotes. So let us try to see what are those additional complexity in the process of transcription which takes place only in the eukaryotes. Like the very first complexity is that there are at least 3 RNA polymerase in the nucleus, right? So the RNA polymerase 1 transcribe rRNA. The RNA polymerase 3 is responsible for transcription of tRNA, 5 srRNA and snRNA. And the RNA polymerase 2 transcribe precursor of mRNA, the heterogeneous nuclear RNA. Right? And the second important complexity is that the primary transcript contain both exon and intron, which are non-functional. Now what happened? We already spoke about them, right? So it has been observed that intron in those regions which do not code for any protein, nobody want introns because in any ways they not code for proteins, so they need to be removed. Now the question is, the more complex organisms have more amount of introns are found. So it is very simple organisms, it is possible that there is no introns, like for example in the eukaryotes. But in the eukaryotes, the primary RNA transcribed are non-functional due to the presence of introns. Then the process of RNA splicing is there, and by the process of RNA splicing, introns are removed. Now this non-functional mRNA becomes functional and mature mRNA has been formed. So children, there are the couple of these additional things which had to take place in the case of eukaryotic RNA. Now children, it has to actually reach the ribosome. Now before it come out of the nucleus, it has to undergo few modification, and these modification includes capping and tailing, right? So let us see what are they. So children, additional modification to the 5' end and 3' end of the eukaryotic pre-mRNA. So what kind of modification are these? That is known as capping and tailing. What is capping? Capping is the addition of a cap to the first nucleotide 5' end of the pre-mRNA, right? So pre-mRNA के पहले nucleotide के 5' end पर cap का attach होना. So as a result, pre-mRNA will be formed, right? So as soon as pre-mRNA is formed, the 5' end will have the cap-like structure. Now what is this cap which is just being added? In fact, cap is the first modification that takes place in the initiating form of mRNA. Now children, what do we mean by capping? What is this capping actually? Children, this ensure protection to the transcribe. Now this is the initial mRNA, and it has got the very important sequences which is going to decide the sequence of amino acid, and that is how it is going to help in protein synthesis. So this information which is there, this mRNA is extremely important, so it need to be protected, right? तो capping actually होता क्या है? Cap is nothing, it is formed by methyl guanosine triphosphate, right? तो methyl guanosine triphosphate is being added at the 5' end. Tailing की अगर हम बात करते हैं, so tailing is actually the addition of polyadenine, right, tail to the 3' end of pre-mRNA. Multiple adenyl group add हो जाते हैं इसमें 3' end पर, so that process is known as tailing. Okay, so now it is ready to move out from the nucleus to the cytoplasm. So these are some of the complexities seen in the process of transcription. I hope you got each the concept clearly. So till then, have a nice day and thank you.